Welcome to the SAN Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, please take a moment to like, subscribe, and share this with your friends and family. It really supports the SAN Science Experience and helps us continue exploring cutting-edge research. Okay, let's unpack this. We are diving into a truly surprising piece of research today. It sort of redefines how we think about cancer cells surviving in the toughest place they can be.
The tumor microenvironment.
Yeah.
The TME.
Exactly. And the whole story really starts with a paradox. The TME is it's hostile, it's starved of oxygen, resources are scarce, competition is fierce.
Right.
So by all logic, the cellular power generators, the mitochondria, they should be just sputtering out, shutting down.
But they're not.
Not at all. In fact, aggressive tumors and the immune cells that actually help them show this incredibly robust metabolic activity. Yeah. So how? How are they powering this whole system?
And that observation is, it's profound. It points to some kind of unknown mechanism that's sustaining this whole, metabolic ecosystem. And this paper reveals that cancer cells aren't just passive scavengers. They're not just fighting to survive.
No, they're actively orchestrating a massive resource-sharing program.
That's it.
And the central, really game-changing finding is that cancer cells act as these sophisticated metabolic hubs. They're hubs of intercellular mitochondrial redistribution.
Which means they take mitochondria, the energy factories, from healthy host cells, they process them, and then they actively relay them to other cells nearby.
It's a form of metabolic control. They're literally deciding who gets energy and who doesn't.
They're controlling the entire energy grid of the microenvironment.
So, okay, let's follow the evidence. How do we know for sure that these mitochondria are actually moving from cell to cell?
The researchers use some really reliable tools, specifically fluorescent tracers that are designed to light up mitochondria.
So they glow.
They glow. And when they tagged healthy immune cells with this tracer and then introduced them to tumor cells, they saw it happen. The cancer cells rapidly just soaked up the glowing mitochondria. The transfer was happening.
Okay, so acquisition stealing energy, that part makes sense for a cancer cell. But the really surprising part is the redistribution. How do we know the cancer cell is the middleman, the hub?
That's the critical experiment. They proved it. The transfer between two different types of immune cells, it only happened when a cancer cell was physically there, in direct contact.
So the cancer cell is the required intermediary.
It's the control point. And this isn't just in a lab dish. They confirmed this in human cancer patients using computational analysis. They could see mitochondria moving from the fighting cells to other, more supportive immune cells.
This is where it gets, just fascinating. What happens to the mitochondria once they're inside the cancer cell? Do they just sit there in storage?
No, not at all. And that's the key. The vast majority, we're talking over 80%.
80%.
They actually fuse seamlessly. They become part of the cancer cell's own mitochondrial network.
Wait, so the cancer cell isn't just passing along isolated units. It's transferring a composite, a blended network of its own material and the stolen stuff.
Precisely. A hybrid organelle. They confirm this genetically by tracking a DNA marker unique to the cancer cell. That marker showed up in the recipient immune cells, but only if they had also received the external mitochondria.
Wow. So it's blurring the genetic line between itself and the host.
It is. And the delivery method for all this.
The high-speed transit system.
It's primarily through these thin, actin-rich projections. They're called tunneling nanotubes, or TNTs.
TNTs.
Right. And when they blocked the formation of these TNTs, the transfer it stopped. It dropped by over 85%. These are the main highways for this whole energy economy.
Okay, let's pivot to the consequences.
Yeah.
What happens when an immune cell gets this hybrid energy payload? Because the paper shows the effects are very selective.
Highly selective, and they all favor the tumor's progression.
This is the core immunosuppressive strategy, then.
It is. The transfer selectively fuels the immune cells that protect the tumor, and at the same time, it drains the cells that are trying to fight it.
So let's talk about the ones it helps first, the suppressive cells.
Right. So for cells like neutrophils and macrophages, getting this energy boost pushes them into a highly pro-tumoral state. For example, neutrophils start forming these sticky web-like structures.
Yeah. Nets. Neutrophil extracellular traps.
Exactly. And those nets actually help the tumor survive and spread.
And crucially, this transfer also promotes the expansion of TREGs, the regulatory T cells.
Yes. TREGs are the immune system's brakes. So by giving them this extra mitochondrial fuel, they become way better at their job.
TREG is stronger.
Much stronger. They produce more suppressive factors and get much, much better at shutting down the anti-tumor T cells. The tumor is strategically upgrading its own security detail.
So what about the cells we want fighting? The CD8 plus T cells, they also get these mitochondria, but the result is the exact opposite.
Right. It's the opposite. For them, receiving this cargo drives them into a state of profound exhaustion.
They just burn out.
They burn out completely. They start showing all the classic markers of fatigue, and critically, their total metabolic capacity just plummets. Both their mitochondrial respiration and their ability to use other fuel sources.
That is just a devastating two-pronged attack. Fuel your guards and induce total metabolic burnout in your enemies.
And the evidence really shows this is a central driver of why the immune system fails. When the researchers blocked the transfer, tumor growth slowed way down, and the number of fighting CD8 plus cells went up.
So we've seen how this creates an immune sanctuary. But let's go back to the cancer cell itself, the hub. Why go through all this trouble, the fusion, the redistribution? Why not just hoard all that stolen energy?
Because the fusion process isn't just about sharing. It's about optimizing its own internal fitness, specifically its ability to build things, macromolecular biosynthesis.
How does mixing its own mitochondria with a neighbor's actually improve its internal factory?
It's an architectural gain. When the external mitochondria fuse, it triggers a physical, structural change in a key metabolic enzyme inside the cancer cell.
And this change?
This change basically flips a switch and boosts its production efficiency.
So the stolen goods are actually a tool to remodel its own machinery, making it a better, more productive cancer cell.
That's it exactly, and they proved it. If you block that initial fusion step, the cancer cell doesn't get that specific metabolic gain. The architectural change is key.
And this whole mechanism also seems to solve another major problem for the tumor.
Resource management. Absolutely. That TME is a harsh place, and some of the stolen resources might be, damaged or suboptimal.
Right.
So the researchers tested this. They had cancer cells take in mitochondria from immune cells that were deficient in complex 1, a critical part of the energy chain.
So they fed the tumor broken equipment. And did it still work?
It did, which is amazing. The transfer and fusion of these broken, complex, 1 deficient mitochondria still improved the cancer cell's overall energy status, just as much as healthy ones did.
So the fusion process lets the cell refurbish or salvage value, even from junk parts. It's preventing a catastrophic energy failure.
It makes the entire system incredibly resilient.
So bringing this all together, what does this mean? This research fundamentally redefines the cancer cell. It's not a passive scavenger anymore. It's an active, organized distributor controlling the entire energy economy around it.
This idea of organelle sharing is it's deeply significant. It really evokes that foundational moment in biology, the symbiosis that created our own cells.
Mitochondrial symbiosis.
But here in the TME, the sharing isn't for cooperation, it's a weapon. It reinforces the tumor's strength, and it builds this powerful immune sanctuary, all by blurring the lines between the tumor and the host.
So this discovery, this whole redistribution mechanism, it's like a new central organizing principle of tumor biology. It opens up completely new therapeutic avenues, things totally separate from our current strategies.
And it raises a really important question for you to think about, especially with modern immunotherapies. Given how damaging this is to T cells, could this exact mechanism be one of the reasons that advanced therapies, like CAR-T cells, get so exhausted so quickly when they enter a tumor?
Something to definitely mull over as this field moves forward. That was a truly deep dive into the tumor's hidden energy economy. If you enjoyed learning about this exciting research, please remember to like and subscribe. Your support is vital to the Sands of Science experience. We'll be back soon with another exciting paper.